systems here. We spoke about an organism earlier, but everything trying to interconnects. Now, type 5 secretion is a particular favourite of Ian Henderson, who some of you may have met, who works here as a bacteriologist. Now, the most common kind of type 5 secretion is the autotransporter. And what happens here is you have a signal sequence that targets the protein to the sec translocon. That goes through into the periplasm. And the autotransporter has a number of different domains. There's an N-terminal, so-called passenger domain, which is what will actually provide the functional activity of the protein when all is said and done. Then there is a translocator based at the C-terminus. And this C-terminal translocator enters into the outer membrane and forms a beta barrel. And in so doing, it provides a pore through which the rest of the protein goes. This N-terminal passenger domain goes. So the beta domain forming a pore here, the passenger domain here in the periplasm, then gets threaded through from the C-terminus towards the N-terminus. It folds up as it comes out the other end, and you usually end up with a system where the protein is tethered to the outer membrane as one complete covalently linked protein, with the beta domain in the outer membrane and the passenger domain free. Now, sometimes the passenger domain actually then gets cleaved off and becomes soluble and goes off into solution as well. And there are many examples of autotransporters in pathogens. There are now hundreds, there may even be thousands of them that have been catalogued in encoding bacterial genomes. But they encode things like adhesins, degradative enzymes, cytotoxins. One example here, Neisseria gonorrhoeae has an IgA protease that is secreted through an autotransporter route. So this protein specifically targets the IgA immunoglobulins in body fluids, destroying them and thereby removing a barrier to the growth of bacteria and colonization of those surfaces. Now, a variation on the theme of the autotransporter, another kind of type 5 secretion, is the so-called two-partner secretion. And effectively what you have here are these two different functions we mentioned already, separated out into two separate proteins rather than two separate domains. So you have this protein TPSB forming a channel in the outer membrane, 500 to 800 amino acids long, fairly conserved, and actually shows homology with the equivalent domain in the autotransporter. Then you have the secreted protein, and these can be extremely large proteins, over 3,000 amino acids at one extreme. They have this so-called TPS domain at the N-terminus, which allows them to interact with the secretion system going through the beta barrel, and they typically form beta helical structures and fold up outside the cell. Once they get through this pore formed by the beta barrels, they then form these complex arrays of beta helices outside the cell. And there are a large number of these also now being catalogued. Here are some of them listed in Haemophilus influenzae. We have these so-called HMW adhesins, which mediate adhesion. In Bordetella pertussis, filamentous hemagglutinin, one of the major virulence factors, which is involved in mediating adherence to host cells, goes out this way. And then we have Serratia marcescens, producing a hemolysin using this approach, using this mechanism. Okay, so that was type 5 secretion in a few moments. Now we need to just quickly consider type 2 secretion. This is quite common in gram-negatives. It's got important roles to play in virulence and physiology of the cell. So all sorts of enzymes get secreted this way. In Pseudomonas aeruginosa, inside, say, the cystic fibrosis lung, it's secreting elastase using this approach, using this mechanism. Many toxins go out this way. We already mentioned ETEC when we and the heat labile, the LT toxin. We have also cholera toxin being secreted in this way. There's a spelling mistake there, I should say biogenesis, role in pilus biogenesis. So there are commonalities between this secretion of these proteins and the production of some kinds of pili. And again, this is a two-step process, as with type 5 secretion. We have the sec or TAC pathways across getting stuff over the inner membrane. Then we have this type 2 secretion system getting things across the outer membrane. Now we actually have quite a complex protein architecture here when we look at the type 2 secretion system. So in the outer membrane we have this complex, we have a protein known as a secretin, which is actually forming a pore in the outer membrane and allowing things to go through. There's a pilotin, which is actually involved in targeting things to be secreted. There are these so-called bitopic inner membrane proteins, or BIMPs, which are interacting with the secretion apparatus. They're stuck in the inner membrane and they're basically facing in both directions, towards the periplasm on one side, towards the cytoplasm on the other. And then you have this, and you also have this secretion dynamic associated SDA protein interacting. You have in this inner membrane complex, as well as these, you have these platform proteins and this ATPase. So again, as with other systems we mentioned, there's ATP being hydrolyzed to actually get proteins across this system. So the so-called platform proteins, highly conserved, interacts with these BIMPs and with the ATPase. We don't quite know what it's doing. It's part of this complex, and it seems to be important, but quite how it all fits together is not clear. Now, I mentioned these bitopic inner membrane proteins. They're linking the cytoplasm and the periplasm together, spanning the inner membrane. They're probably also interacting with cytoskeletal elements in the cytoplasm there. And there's the ATPase. And at its most basic, there's just one of these in type 2 secretion systems. Some other systems that are more elaborate might have more of these, and we'll say more about type 4 pili in a moment. The secretin in the outer membrane, I already mentioned. The best characterized one of these is probably a protein called PUL-D from Klebsiella. GSPD is another one that's been characterized, fairly highly conserved. And you get this channel in the outer membrane. There's this ring-like structure of the lipoproteins coming together to form this pore. And the pore is sufficiently wide that you can get folded proteins through it. So if you remember, we said that you get things like LT assembling into the holotoxin in the periplasm and going through a pore like this. There's a kind of gating mechanism as well. So part of the end's not shown on this figure here, but there's an N-terminal domain that sort of swings round and closes the passageway until such time as a protein that's going to go through it is ready to be engaged, and there's some signal transduction that allows this to happen. So that N-terminus interacts with secreted proteins, and we get, so here's cholera toxin coming in, and we've got this possibility of interactions involving these beta strands, particularly with proteins that already have beta strands coming in here, and this kind of crosstalk between the secretin and the secreted proteins in that way.